Miss Berkers here. I just wanted to go over some quick misconceptions from the last lesson before we go on to lesson 8 so that you don't get lost along the way. So the first thing I wanted to point out is the difference between an organ and an organ system. So when we move up in the level of within the body, all parts of your body are made from atoms, which these atoms then join together to make molecules. These molecules then make your cells. So this was the base level that we were talking about in the lesson, was the cells. Cells then come together to make tissues. So tissues are a group of cells that has the same job and that works together. So something like heart and muscle tissue is an example here, or something like white brain matter would be an example of a tissue in your brain. These tissues then work together in it to make an organ. So for example, your heart is an organ, your brain is an organ, your liver is an organ. So there's more than one type of tissue in every single organ. But then groups of organs come together to make an organ system. And so this is the part that threw some of you off in the quiz. So when it asks which organ system coordinates movements in the body, it's not looking for your brain, because your brain is an organ. But what organ system is your brain part of? That would be the nervous system. So when you're reading the question, just take an extra second, read it carefully, and double check if it's asking for an organ system or just an organ. So that was one question that a lot of you struggled with, and I think it was just, if you take your time, read the question carefully, you'll be able to see that difference. And then the microscope. I know we didn't get a chance to use the microscopes in class, so that does make it a little bit harder, but it will be something that we do once we are back in the classroom and able to be doing that way. So you will get a chance to do that. One bit was labeling some of the parts of the microscope. This is something that you will need to be able to do. So just take some time, re-go over the diagrams and make sure that you've memorized the different parts. One here in this diagram is the eyepiece. 2 is the stage. And 3, in this example, it is a mirror, but you can also just go a light or the light source, because in all the microscopes that we will use at school will just be a light one that you plug in. So, and then there was one question that threw a lot of you off that was actually a really tricky question. And it was, what would you turn in order to move the objective lenses up and down? So most of you have had the chance to use the microscopes at school, and it's not the objective lenses or these bits here that move up and down, but the stage that moves up and down. But here you see that the rough focus knob is closer to the objective lenses than the stage. So it's actually the objective lenses in this example here that goes up and down. But you always turn the rough focus first. And it's only once it's mostly in focus that you then turn to the fine focus. So if it doesn't ask specifically for it, always talk about the rough focus first and only use the fine focus once it's in focus from the rough focus. So if you're, for example, before you put a new slide onto the microscope, you always want to make sure that the stage is down or the objective lenses are up, so they're as far apart as they can be. So when they are as far apart as they can be, that's when you use the rough focus, that you just move it all the way up or all the way down, depending on what you're moving, so that you're able to have as much room as you need to put that slide in there before you go on to the next step. So if you're just moving it without trying to focus it, you use the rough focus knob. So hopefully that clears those bits up for you. Now I'd like you to go in and try lesson 8.